First of all, welcome warmly to the second VAAM Industry Academia panel. My name is Christine Lang, and together with Stefan Pelzer, we are your moderators or hosts today. And we are very happy that we can welcome you here for a seminar on two different aspects coming from industry and academia, just as the title says. And today we will have talks on strategies in microbiological diagnostics. The two speakers I will present in detail later before they talk, and you can see here their names and where they come from, even if you want to read their the titles. But before we go into this topic deeper, I have two announcements to make. One is, one is that we would like to bring your attention to the second time that we are awarding the VAAM Innovation Award. This award will be given to someone from industrial research who has been there for industry labs like startups or large-scale industry, and is known for outstanding scientific work in the field of microbiology. You may remember that the first time we awarded this prize was in last year's conference, and the next time, we said it's a 2-year period, will be next time when we hopefully meet in person in the next VAAM annual meeting. And please just look if you know someone who would fit into this award, and then think about nominating him or her and send the nomination to the VAAM. The deadline is end of the year, so you will hear this a second time next time. Also, what I would like to make an announcement of is that this is the second, but the third session and the third round of the VAAM Industry Academia panel is already planned for in 2 months' time. This is the 15th of September. And we also know the topic by now, it's microbial metabolomics. So we sincerely hope, and I just want to say this in the beginning, where we are going to. Now let's come to today's seminar and session. I would like to introduce the first speaker. Before I do this, there are two technical things. The first thing, you may have seen that we are recording this session, this panel. So just that you know about that, you can later on have a view again on this in the website. And the second thing I would like to point out that we will have the two talks. In between two, there will be a short time for discussion, and afterwards, the second, we will have more time for discussion. So we welcome your questions in the chat, in the chat function. And Stefan today will be the moderator to look at the questions and see that. So more or less enough me. I'd like to introduce now Jiri Snaidr, who is the first speaker in the panel. Dr. Snaidr is the CEO and founder of the company Vermicon, and this company is already founded in 1997. So it's a 25-year jubilee year this year, which is quite a great achievement to have a company for this long time. Before he started the company and what he's doing, he will tell us in a few minutes. He did research on non-cultivable bacteria and detection of microorganisms at their site of action. And he was part of the research team who developed the fish technology. I warmly welcome Jiri Snaidr, and Jiri, the floor is yours. And I stop my slides, and please set up yourself.
Yes, thank you very much, Ms. Professor Lang. So I'm very happy to be able, just, okay, it just vanished my presentation, no problem. It's okay, just Just a minute. Okay, now it's here again.
Perfect.
So you
that
see it?
looks good.
Okay,
Yeah,
perfect.
that's it.
So, so I'm very happy to have a talk today about microbiology and about direct microbiology. And yes, what you said, it's 25 years. It's really a long time. And really today it feels like still being a startup. So it doesn't really feel like 25 years, more or less like 25 months. Yeah. So today's talk is about using direct microbiology in industry and how an unbiased look into samples changes our perspective. And the first is, so we have to ask ourselves, what is direct microbiology? So when we look at the good old cultivation and the quotes about the cultivation, so that's the way microbiology has worked for centuries. Robert Koch, and only growing means knowing. I really like this one. But the problem is a little bit with the cultivation is that is well, it's really old, about 130 years now, 140 years, yeah. And today we rely mostly on the methods of yesterday, so it's not a direct method. So the other one, the beloved and established PCR with malice, quotes are PCR is super specific, it's so easy. And I really have to remember here that we talk about industry. So what we are doing are applications, we are developing services and products for industry. And we do not talk about really high-tech equipped professional lab in the university, but we talk about the German Mittelstand, I would say. Yeah. So it's not always that easy because you need some very high-equipped rooms for the PCR, and it's also the question: do you really know what you are amplifying? So it's not a direct method. The other one is the mother tool of all databases, sequences, the quotes. It's got so inexpensive. That's true, because I remember so several years ago when I made a sequencing analysis of the 16S RNA. It was room. It was enough for a whole diploma thesis. And today you pay, I think, several euros for a sequence. And also here often partial sequences for analysis for phylogenetic are enough. And hey, it's okay, 300, 400 bases, it's okay. No, it's not okay. It's not okay to do really a profound phylogenetic analysis with partial sequences. And this is also the reasons why the databases get really worse and worse. And the question is here, are microorganisms just sequences? And again, it's not a direct method. Okay, then all method with omics, metagenomics, metabolomics, and so on. And the quotes are here: we get all data we need to know. This is so fascinating, and it's a fantastic look into the communities. Well, I would deny it. It's not a look into the communities. These are just data. And again, it's not a direct method. So when we ask ourselves, what is direct microbiology? So there are two very important words. The one is direct, so direct microbiology, not indirect microbiology. And the next, and this is very interesting because the next is sometimes forgotten. We talk about microbiology. We don't talk about just data. And what is not microbiology? And often today when I see that, people are forgetting this. Microbiology is not just data. Microbiology is really more than that. And the same as this comparison, humans are also not just data. You can classify them regarding religions, ethnicity, name, profession, etc., etc. But humans are not just data. I would really say we are more than just a bunch of data, and so are the microorganisms. Well, microbiology, not data of microbiology, not data of microorganisms, but microbiology is always microbial. ecology. We have always a huge network of interacting different species and population. It's not that we have a sample and there's a Salmonella, a Listeria, and Legionella. No, this is a complex biocenosis. It's a complex biocenosis, and they are in constant interactions. So the problem is the reality of the microbial sample is always a puzzle and can never be obtained by mere indirect microbiology. So we often think when we apply indirect microbiology or indirect methods that we know what the reality of the sample is. We know what the reality is, but we really don't do this. Yeah, this is, and what we think to know is not necessarily what the reality is. And this is simply what we call the gap. And our vision is closing the gap. So let me show this in this diagram. So what we think is all we get from the indirect methods, metagenomics, PCR, cultivation. And on the other side, we have the real reality, I would say. And the difference between this is the gap, because we think what we know, and on the other sides, we have the real reality. But there's a difference. This is the gap. And our vision and our aim is to close this gap with direct methods, so that we combine this direct method with indirect methods to get really the reality of the microbial sample. So for us, direct methods are the missing piece for the puzzles. And these are all methods which allow the direct in situ access of information. And these direct methods allow the exclusion of bias. They allow the detection of all microorganisms. They allow a real quantification. They allow the correlation function of identification. And what's very important, to analyze biocenosis. And every microbial sample is always a biocenosis that allows spatial analysis. So, and this is very interesting, because here is a look into the fish sample of a colon, yeah. And you see here the fish probes for Firmicutes, yellow, for the Bacteroidealis in pink. And now you have the whole picture, because now you have a direct look, with a telescope. So you have the direct look into the sample. So what is a direct method? Very, very easy. A direct method is a sample, and on this sample, you apply direct method, and this reveals in situ what's inside. It's very simple. You have a sample and you want to know what's inside. You don't want to know what's inside using a specific technology. You don't want to limit yourself. You don't want to say, okay, when I use this technology, I can analyze this. No, it's just a sample, and you want to know what's inside. That's easy. It's easy. It's like a room. There are a group of people, and you want to know what people are inside. What people are they inside? So, and for us, the first sense in situ hybridization is the most important direct method. And the pillars of the first sense in situ hybridization, I'm sure that you know, is it's on the one side phylogenetic analysis. Then we can do a development of the specific probe. We apply them to the sample and get the results. We get the shining bacteria in the sample. So this is summarized. If you apply it, so in the headlines, gene probe technology, because we made a further development. We made a further development of fish. We developed it to an industrial standard, and so we call this fit gene probe technology. But it's based on the fish technology. So what happens with fish? Gene probes contact the sample, and inside the samples are different cells, and with the gene probes we have specific probes for bacterium A, which are labeled in green, specific probes for bacterium B, which are labeled in red. And then all gene probes penetrate all cells, but only inside the cells where they find the specific targets, they really bind. And so the non-specific gene probes are washed away, and the cells begin to shine. This is a nice example from a probiotic yogurt. You see the phase contrast, and with phase contrast, with the Microscopy is also a direct method because you're looking into the microscope. But the problem, you know, is with the microorganisms, they are rods, they are cogs. Sometimes they are a little bit other shape, but it's not that easy, or it's impossible to analyze the bacteria due to the shape. And by applying the fish technology, you see here the yogurt producing bacteria, the Streptococcus thermophilus in red and the Lactobacillus acidophilus in green. By the way, this a probiotic yogurt, and you know, in the last, I would say 10 or 20 years, it got very famous, probiotic yogurts. But well, I would say the best with the probiotic yogurts is the marketing. Marketing is great. I love it. Yeah, it's always happy people, healthy people, and they eat the probiotic yogurt and feel more and more better. But I would really suggest, I would advise you, if you really want probiotic bacteria, so go into the pharmacy, go and buy some probiotics, some real probiotics, and get them. Because the problem is with the probiotic yogurts, often they are in low amounts and not viable anymore. But marketing is super, and I have to admit that I'm eating probiotic yogurt too, because I love the marketing. Well, fish allows then the specific detection, real quantification, measurement of activity, and it allows differentiation of dead or alive, and it analyzes the association interactions between populations and allows also spatial analysis. So coming back to vermicon, just 2 slides. So what we are doing this quarter of century are direct microbiology for the industry. And so we are coming from the technical university, from the Department of Microbiology. And we see ourselves as pioneers of fish technology because we were the people in the lab who applied first the fish for visualization and identification of bacteria. We are GMP and ISO certified. And yes, of course, now we have decades of experience in industrial microbiology. We are located here in Hallbergmoos in the Munich Airport Business Park, very near to the airport. So I'm see here the airplanes going up or not down. And we see ourselves as a specialist for microbial ecology, pioneers of modern microbiology, diagnostics, and also experts for, of course, fish and biocenosis. And our portfolio are services for the detection analysis of microorganisms, products, and of course, R&D or consulting or projects. So, but let me get a step back. Let us make a short change of perspective. Let us look at the gold standard. And here we are proudly presenting the gold standard, cultivation. So this is interesting because cultivation is indeed today the gold standard. So, and I would say the gold standard should be the best method, of course, because otherwise it wouldn't be the gold standard. So really? Really? Well, let us look. Let's make a change of perspective. Let us imagine that we have a mixture of modern methods, and these modern methods are the current golden standard. So we live in a century where these modern methods are the golden standard. And now comes a new kid on the block. And this new kid on the block is called cultivation. And this new kid on the block says, hey, I'm so cool. I want to be the new gold standard because it's great. It requires a medium to transfer bacteria out of the sample. And it's so cool because it needs different media for different bacteria. It's absolutely great because, don't look at this so close, because only 1.0 to 1% of all bacteria grow there. Yes, it's really so. Up to 99.9% of all bacteria cannot be cultivated with artificial media because it's too complex, the population. And it can't quantify correctly because of mixed cultures. And it proves only indirectly whether there are bacteria in the sample or not. And it can't show populations or single bacteria, and it can't show associations between bacteria. But hey, come on, let us be the gold standard. So why on earth would you trust that method? Why on earth? But it's interesting. Today, it's the standard. It's the gold standard, the cultivation. And And what is really interesting is that every new method must compare with it. And imagine now you have the cultivation from a, I would say, quality level here, and you have a new method which is really, really better. But the better the method is, the more difficult it is to compare it with the cultivation, of course. And so if it's not possible to compare it, the new method has no chance to be an alternative for cultivation. And I would say really, this is crazy. This is really crazy. And I would say cultivation has just two single advantages. The first, yes, that's great. It delivers biomass. It delivers living bacteria. They grow on a fish or on a media. You can do what you want to do with this bacteria. And the other advantage is it is old. It has a huge lobby. Because when we look at the cultivations, we have big companies who are deep in the business of media. Most of the regulations, and believe me, we are in several industrial segments like food, beverage, water, wastewater, pharma, and so on. We know this regulation. Most of the regulations are based on cultivations. Almost all product releases of the industry are based on cultivation. Certifications are based on cultivation. The whole bureaucracy, whole Berlin believes in cultivation. I had several letters with Berlin explaining the disadvantage of cultivation. We have people with the experience of cultivation. Yeah, it's always what I've done, and I remember also in the university I've done cultivation. It's cool. We have quality control. It's based on cultivation. We have safety measurements based on cultivation, and this is a huge advantage, because if we would cancel cultivation, this would be like an atomic bomb, a huge explosion. Because let me say it, if you cancel cultivation, so this is a smash. This would be like the big bang of microbiology, because all these safety measurements, human experience, certification, bureaucracy, regulation, product release, and so on, there then need changes. And let me say, this is difficult. So don't forget, it's not about the best method, not only in microbiology, but also sometimes in other segments, but it's about the spider's web. It's about the spider's web. It's about this web, what I've shown, so of this old cultivation method in the regulations, in the certifications, and so on. So believe me, I've tried for 25 years now to kick out the cultivations out of the certification. I showed study after study it's better with this and this and this modern method. I don't say only fish, but also PCR and other methods. But it's almost impossible. And now I always said, when I retire, maybe I will see that the cultivation method will not be the gold standard. I think I will not survive it. I will maybe the next or over next generation, but I will not see that this gold standard will be cancelled. So some examples for using direct microbiology in industry and how it allows a direct look into the samples. So one side is wastewater. Wastewater, when we look at wastewater or biological wastewater treatment plants, so the bacteria are really the engines. And so what we are doing is, so we are trying to improve the wastewater, the biological wastewater treatment plants by switching or by analyzing and switching the populations which improve the degradation performance. We are analyzing the nitrifications. We are analyzing the pathogens in wastewater treatment plants, the biological phosphorus elimination, denitrification, bulking in floating sludge, anaerobic reactor. So in principle, the whole wastewater is just a wonderful paradise for bacteria. I would say it's the last paradise. This is a paradise where until now the apple has not entered the room. So, and these are some examples from analyzing wastewater with fish, so detection of archaea bacteria or detection of nitrifiers. And there you see really nice, this close association of the different populations. And another example is food and beverage production. Yes, you have again, for the end product, you have this regulations where they say, okay, you need this and this number of colony forming units. But as I said it before, a sample is more than that. And just a small story. When I started vermicon, so I went to the food industry and said, hey people, this is so cool. We know that you analyze Salmonella and Listeria, but now we can analyze the whole bunch of bacteria, all bacteria which are in your sample. And the managers looked at us and said, well, thank you very much. Here's the door. Thank you. Of course, they didn't want to know what's still in their product, but they had just to fulfill the regulations. But of course, they have always problems. There's always problems in process flow with the raw material, and with the direct method, it's possible to have a direct look. To have a direct look. And real microbiology, applying real microbiology in this process means monitoring all bacteria, analyzing the population dynamics, and acquiring knowledge about what really happens, not just about the 1, 2, 3, 4 bacteria which can be cultivated. So this is an example for Acobacter analysis in meat, or we do also the detection of Alicyclobacillus, which is a contaminant in drinks. Here's a close association, or as a symbiotic association of Alicyclobacillus and yeast. And also what is really fascinating, we're doing a lot of work in the microbiome. This is really a wonderful network. And over the last 10, 15 years, we've learned so much about the microbiome, but still at the beginning, we know that the microbiome so much influences our health. And we have about, I would say 100 billions of bacteria. So 100 or 10 more than human cells, and this is one complex biocyanosis consisting of many different smaller biocyanosis. For example, you know it in the mouth, you have almost so much bacteria in the mouth like people on the earth. It's dynamic, complex, a huge genetic pool. And for this analysis, a direct method is perfect, because this is an example from or a graph from a colon, and you see there on the left side is health state, on the right side is the disease state, and you have these scripts. And normally in a health state, you have these, I would say, just good bacteria in the crypt, and they're protected by the mucus, but they are bad bacteria, sorry for the simplification, good and bad bacteria, which I would say, which degrade the mucus. And when the mucus is degraded, so the bad bacteria can grow in the crypts and get rid of this good bacteria. Also, we have attacks of the pathogens with bacteriosigns, and also we have, when you eat single sugar or complex sugar, with a single sugar, you are in favor of these pathogens. And also we have pathogens which influence the flow of the fluid in the colon. So, for example, the Vibrio makes this, the flow increases and the good bacteria washed out, but the bad bacteria, which are flagellated, stay inside. So we have really a lot of interactions with cause constant changes. And when you just look at the data, when you just look at the sequence data, this is not enough, because in these cases, the sequence data will, in many cases, not vary. But what will vary is the spatial analysis. So, because this is microbial ecology, this is not just data. And it's often the question, population dynamics, it's cause or effect. Yeah? So we have the sequence data distribution profile, but with the fish analysis, we see the spatial distribution, we see the real quantification, and we have just more information due to this additional fish data. We have the analysis of relationships, and it allows a better assessment. So coming to the slide, what you've seen, so about the humans, when we look at ourselves, just in data, religion, ethnicity, we are more than just data. So, but when we look at ourselves and do a spatial analysis, we suddenly see something other. We see the whole world, we see us. We see ourselves. We get the whole picture. And I would say this whole picture is so important. Please, I really, after 25 years, let me advise you, always look for the whole picture. Don't get lost in this data. Look at the microbial ecology and change your perspective always. And with this last sentence, change your perspective, which can be applied not only to microbiology, but also often to other fields of life. I want to thank you very much for your attention. Thank you.
Thank you very much. Great, fascinating talk. Stefan, over to you. Maybe we have time for 1 or 2 questions at this point.
Yes, thank you, Christina. Thank you, Jiri, for this really interesting talk and the insights from your research. I can't see any question in the chat yet. I hope it's not connected to my. There is a question from Mamdul Hassan. Nice talk. What about detecting new species of bacteria? And what is the target of your probe?
Yeah, so the target of our probe is the RNA. And of course, a new species, this is a very good question, because always the people say, okay, you design the probes from the sequences. So first you need the sequences. Yes, I would say 50%, that's right. So when we have the sequences, we can design the probes and get back in the sample and see the whole picture, because from a sequence analysis, the quantification, please don't rely on the quantification of sequence analysis, because there's always a bias then. So sometimes the people come to us and say, hey, this is amplicon analysis. This is the real quantification. No, it's not. But on the other side, so we can then develop with a new species from the sequences. But on the other side, we have this approach where we apply, for example, 10 group probes, which really cover all the bacteria. And then we see there's a new bacteria in this one group. And then we can go deeper and deeper in this group to analyze this bacterium.
Thank you. There is no other question, so please allow me one other question. I think we are a little bit pushed by the time, but allow it to me. The selection of methodology is always dependent on the purpose, I guess, you have for the analysis. And if it's about speed, which is sometimes also decisive in some contexts, in diagnosis or whatever, how would you compare the fish technology, the direct methodology, with indirect methodologies, and how long would it take to get a result?
Okay, so for me, my most wonderful example is it was in the year 2000, and I visited Australia, and I visited a beverage production site, fruit juice, and they said, well, I wanted them to sell E. coli, and so they were not interested. And then they said to me, hey, look, we have a problem here with a germ. And we are waiting 14 days with incubation, conventional incubation, 14 days. This was interesting. And then I said, okay, which bacterium is that? And they said Alicyclobacillus. Well, we can analyze this Alicyclobacillus with the fish technology in principle within 3 hours. Yeah. So, and for us, it was always important to have a very speedy method, but not to lose any specificity. So we always wanted this combination of speed and specificity. And of course, we have other bacteria where cultivation does not last so long, but with fish, you are really within hours, you are finished.
Thank you very much. I do not see any other questions, and therefore I would hand back to
I would have
Christina.
a question.
There.
Thank you for the fascinating talk and the fascinating pictures. I have a question in terms of sample concentration. So with your method, you only can microscope a certain volume. There are different methods to concentrate sample volume when you think about liquid samples like beverages or water. So what is the method of choice? Is it membrane filtration at the moment, or is it centrifugation, or maybe more innovative?
Yeah, indeed, the best method of choice is here filtration. So we do a filtration of the sample, of the liquid sample, and then analyze on the membrane the different single bacteria with this method. Yeah. So of course, centrifugation would be also possible, but we prefer the filtration on the membrane.
Thank you.
Thank you very much.
Okay, thank you.
I guess in the interest of time, Christina.
Yeah, thank you very much. Without further ado, I thank again to Dr. Snaidr and turn to Dr. Mathias Hutzler as our next speaker. And I'm very happy to have him here. He is food technologist and biotechnologist, studied at the TU Technical University of Munich. Later on, he was scientific assistant at the Technical University in Munich at Weihenstephan. And since 2009, he is the division manager of the accredited laboratory for brewing and beverage microbiology and the yeast center of the research center Weihenstephan for beer and food quality. And Dr. Hutzler will talk about diagnosis, but from the application point of view, his title is Appropriate Method Mix in Applied Beverage Microbiology. Please, Dr. Hutzler.
Yeah. Thank you, Professor Lang, for the nice introduction. I will talk about the different methods we can apply on beverage samples and beverage themselves. In my lab, we analyze about 15,000 samples a year, all kinds of beverages and water, alcohol-free and also alcoholic beverages with a focus on beer. So also feel free to ask about specific scenarios if you're interested in that topic. So my first question is, where is the journey heading to? So I will show you a little bit statistical data. Where will the beverage industry go to the next years? And I also will talk about challenges, problems, and opportunities for breweries and beverage producers. And one main topic is the sterilization, or let's say the decrease of microbial load. I also will show a few methods how we can, let's say, fight microbes in the process. And also, which methods should we choose, which new challenges we may have in the future, also in terms of new microbiota. So this is a statistic about beverage volumes in Germany. It's the consumption per capita, per inhabitant. And you can recognize that there's a decrease over the last, let's say, 15 years of alcoholic beverages, especially beer. Wine and spirits and sparkling wine, like champagne, they almost remained the same, but beer had a significant decrease. On the other hand, alcohol-free beer increased, and some special beers that are also a new, let's say, microbial habitat for, let's say, beer spoilers, for example. And non-alcoholic beverages are growing. Water is growing, soft drinks or other carbonated drinks, and juices are decreasing. So this statistic is like a mirror of also the health consciousness of our German population. So less sugar, less alcohol, and in the soft drink section, there's also a significant increase of, let's say, traditional soft drinks like high sugar lemonades or Coke and so on. So where is the journey heading to? There are a lot of innovative products. For example, here, this is a Frank Juice, is a company that produces freshly bottled and produced juices and vegetable juices, and it's for a diet. You don't eat, you just drink those products for 2 weeks, and it's like a detoxification. So it's focused on a special segment of consumers. Then this is an Australian product in the left lower corner. It's like a jelly soft drink. So you have a special mouthfeel experience. And as you know here, some non-alcoholic beers that are very successful also because they have a very low calorie load, or on the left hand, a very special beer with tropical aroma and impression. On the right upper position, it's kombucha. Fermented beverage with acetic bacteria and some aerobic yeast, depending on the process. They often have the problem that there's still fermentable sugar in the product after they reached the acid concentration they want to have, and then there is a danger of fermentation and then too high CO2 content, and in the worst case, explosion or blow up of the container. But as Dr. Snaidr already said, there's a big interest in fermented beverages, also indigenous fermented beverages that go to an industrial scale because they have a very good let's say health reputation. Then on the right side, lower side, it's the holy, for example. It's hard seltzer. So this is a fermented sugar base mixed with water and aroma. So lower calories than wine or beer. It's not so successful in Germany, but in the States, it's a big market. But as you can see, there are different niches, and every new product is a new world of its own when you think as a microbe. So you jump into a new environment and you have different concentrations of organic acids, sugars, amino acids, maybe ethanol, vitamins, minerals. It's a specific chemical cocktail. And for every new product, you should think about your microbiological hurdles. And a lot of those new products are very susceptible from a microbiological point of view. And often those startups check as last point of the product development the microbiology. I would recommend to do it first, but we often have customers that are then really, really sad because the micro results are so bad after a long development period. And we always recommend to check your chemical specifications in advance. So if you have a plan, or if you have a blueprint of your product that you want to scale up to industrial scale, spend as much time with your specifications and with a check of your chemical parameters that have an antimicrobial influence, like alcohol, or in this case, these are in the table, these are values of Radlers. So beer mixed beverages with lemonade and beer. You have alcohol, you have hop bitter units that fight against gram-positive bacteria. You have a low pH degree, CO2 over 0.4 grams per liter. Mold growth is very unlikely. Then sugars, protein, and free amino nitrogen. So those are basics that we analyze. And then you also can think about to add additives, preservation substances, or also think about other conservation or pasteurization methods. So that's one of the messages of my talk. Which microbes can grow in your specific chemical cocktail in your own world that you want to design with, or that you already have. Then some slides now about beverage production. So when you produce a soft drink, most soft drink producers use this technology. You have an inline mixer. In this mixer, water, syrup, that means sugar, aroma, or some base, food base, and additives are mixed. Then you add CO2, and then it goes through flash pasteurization. And then it's either filled aseptically, or if the product is not so susceptible, it's filled on normal filler. And then you can have different additives like preservatives, coloring agents, antioxidants, sugar substitutes, flavorings, and so on. This is all regulated here in this European regulation. So you only have a certain portfolio you can use or you're allowed to use. What is more universal is pasteurize your final product in the bottle. So that's often done with, let's say, low alcohol beer or sugar containing products, where heat is not so so much influencing the aroma profile. And there's chamber pasteurizations, so you have steam going through your containers, and a fan wheel and a circulation of steam. The important thing is that you should have a good data control, temperature control, or from our experience, old pasteurizers often have problems similar to an autoclave. If that is not really maintained very good, then you can have problems. Another process, it's the cold sterilization with Velcorin or DMDC. It's basically the same process. It's just that you have before the filler here a dosage with a special unit where you dose this chemical, the DMDC. DMDC later converts into methanol and CO2, and it is active for a certain time in the bottle and kills microbes. So my recommendation in beverage microbiology would be set up an appropriate and intelligent routine microbiology and intelligent specifications. I often made the experience when you go for consulting to any company that you collect so much data and nobody looks at the data. And then you can already see some tendencies if biofilms increase, if you just take a little time and have a look at the data from the last year, and often you can just see something increasing or growing. Then my second big recommendation is, if you have growth or if you have any microbes, identify them and classify them, and also evaluate those positive results. And the third point would be, do a risk assessment of your products, which microbes can grow in those products, and also do challenge tests, inoculate also the microbes you find into your products, and check your house microbiota. Is it adapting or not? So I know this slide, it's very inconvenient, but I just want to give an overview. So these are specifications for different types of beverages: high pH, low pH, with CO2, without CO2, and then some intermediate products like sugars or bulkware, intermediate ware, And these are specifications of my old colleague, Professor Werner Bock. They are still valid. There are other specifications from other associations, but this is good for a first overview. And the spoilage and the common microbiota can be evaluated very good. So if you investigate your raw material, your intermediate product, and your final product and your shelf life samples, you can assess here your results very good. So the next slide wants to answer why shall we identify microbes that we already have on the plates or that we found with other methods. It's always interesting, which spoiler is it? Is it a spoiler, or is it just like a hygienic indicator, or is it a germ that does do nothing? Has it any damage potential for my product, or for an intermediate product? Is it important in terms of hygiene? And can I obtain information? So maybe check databases, check literature, or ask institutes like us or companies like Vermicon, for example. And then it's important, with this information, you also can localize this microbe, the source of contamination, use, for example, specific methods, special media, combined with special real-time PCR system or with the microfiche, and then eliminate the source of the infection or the source of the contamination. And then you also know what you have done. Maybe you also did a reconstruction of a certain part of the process, or you changed the machine, or you adjusted your cleaning and disinfection. And when you do this process, you gain more and more knowledge about your house microbiota. And it's often like this, that every 1, 2, 3 years, it pops up again. And then you already know which microbe you had problems with, and then you know already your countermeasurements. And you know about the harmful microbes if you inoculate them in your product. So that's what we recommend, and you can implement then a monitoring about your house microbiota. So the 2 pillars of classical microbiology, that's cultivation, enrichment, and microscopy. As Dr. Snaidr already mentioned, it's often very laborious and time-consuming, but still the gold standard. And it's always also point how to combine. We have a lot of culture media, very specific, like for example, for super attenuating yeast, like Saccharomyces cerevisiae diastaticus, or very universal one like TSA or PCA, or more for soft drinks like SSL or lactic acid bacteria like MRS. You have to choose the right media for your process, and also think about qualitative or quantitative method. What is the big difference of beverage microbiology to other disciplines of microbiology, like medical microbiology? Often we only have trace contamination. I have here now a pipe system with a schematic biofilm, some green bacteria, so they are often on a certain position, and when fluid runs, they are diluted very fast, and then you only have single germs. Or the other picture should show a yeast propagation tank. You have your cultivation yeast, your starter culture, and then you have a few bugs inside, only a few bacteria. And it's very difficult to cultivate them, or also to find them with direct methods. And that's the big challenge. how can we concentrate on the one hand, or how can we find in fermentation process the non-fermenting microbes. In the middle, this is a biofilm from a water piping system I took a few years in the States. So this would be in between. This is massive microbiological biofilm that shows a big hygienic problem. So during the Corona time, we did a study on fermentation, pure culture plants, so propagation yeast plants where you grow your starter culture for brewing. And with normal methods, you only check for beer spoilage bacteria. They are mostly negative, but we thought, let's check for other microorganisms, and we found a lot. So in 22, of 43 samples, we found cultivable microbes. They are listed above. And when we applied PCR-based methods, it were almost 80%. And those bacteria are also affecting this process because it's an aerobic process. So when you look with other methods, you can still find problems, and that's exactly what Dr. Snaidr mentioned before. If you're only looking with your standard portfolio and you're happy with that, you will probably only find what you're looking for, but there's also some space left and right of this small window. Then there's one crucial point, it's the sample concentration. You can do it with sedimentation, so just leave a bottle there overnight or 24 hours. You can do membrane filtration, concentrate the microbes on the filter, or centrifuge. If you concentrate, then you can apply this direct method, or also other methods like PCR and so on and so on. Okay, I made this very brief. So you have these different kinds of microscopies. Dr. Snaidr already showed here fluorescence microscopy. We in brewery microbiology often use the dark field. It's a very seldom application, but lactic acid bacteria you can differentiate very well because the cell wall is very bright, and I showed here a few examples how bacteria look like brewing contaminations. This is a super attenuating yeast, over-fermenting yeast. You can also check it by a microscope, looks differently. Then we have this massive improvement also in microscopic methods, staining, digitalization, counting systems. You can calculate cell morphologies, use some algorithm. You can connect it with fluorescence, with the IR spectroscopy, or with Raman microscopy. Here with Raman, you can also go into the cell structure and check which chemical compounds are within a yeast cell. With bacteria, we have problems often because the laser is often bigger than the single cell. The spectra are not this good, but it's a nice scientific approach, not yet really in practical microbiology. MALDI-TOF is the method of choice for quantitative samples that are on agar plates. You can very fast identify the colonies. So we have a very broad database for beverage-related microbes. And the two methods that are applied very often in beverage microbiology is real-time PCR and flow cytometry. But you always have to keep in mind, if you take a certain volume, you have your detection limits, like often it's like 50 to 100 cells per milliliter. And it's often a fact of concentration of the sample. If you have issues with unknown germs, for example, unknown microbes, PCR sequencing is like the gold standard. I think everybody of you knows that you use the ribosomal DNA, for example, as target sequences for bacteria and also fungi or yeast. We also developed some typing methods where you can differentiate on strain level. It's depending on your DNA region you use. That's now one method for yeast. If you have different strains of one microbe in your company, so this would be now a differentiation of different yeast strains to avoid cross-contamination or check cross-contamination or do starter culture control. This is often an application we use in troubleshooting. You have beer contaminated and then you check your hygiene of your process. And if you are lucky, then you find the same microbe at another source, and then you have the real proof. For example, in one brewery, we found about 30 different lactic acid bacteria. These are the different numbers. And then in the end, it were like 6 different strains. And then you want to know which one is the one in the bottle or in the bright beer tanks. If you have microbes identified, they are new, then you want to know how they work, which resistance mechanisms do they have, are they resistant against certain preservatives, which acids do they produce. So this was, for example, Lactobacillus cerevisiae that we described a few years ago. And then you know more about your spoiler and the spoilage potential. Does it form off flavors or viscous beer? And that's my take-home message. So every process needs the appropriate methods. And for me, the best is a good mix of standard microbiological methods. You need to fulfill your specifications and have sensible specifications, and combine it with the right rapid method, like, for example, the in situ fluorescence probes of Dr. Snaidr. And that's the exact thing. You need something where you can do troubleshooting, either with external partners like us, or you have already the equipment in-house. And that's my take-home message. A nice picture of an old colleague of mine, of Mr. Engelmann, who visited probably 1,000 breweries or so in his career. And he always said in microbiology, 2 + 2 is mostly 5, in seldom cases 3, but in very rare cases 4. So that means often you have to look very long for a contamination and find the right source, because the source often is a little bit at the beginning of the process, and you detect it later. So thank you very much for listening. That's my last slide. I also want to thank all the audience, the moderators and the VAAM for inviting me. And we do archaeological microbiology, yeast hunting, so also find new microbes, and indigenous fermentations. So that's aside the standard microbiology. Thank you very much. And sorry, it took a little bit longer than expected.
Yeah, thank you very much for your presentation and the insights in this special kind of brewery microbiology. For me, at least, I learned a lot what the challenges are, to be really honest. There's a question, or Katrin, you raised your hand, but maybe this was by accident, or do you have a question? Obviously not. I do not see any question. That's Dr. Snaidr. You have a question.
Dr. Hutzler, thank you very much for the great talk. It was very really good. And I just wanted to direct your attention to a really very good sentence from your side. You said it's every product is a world, a microbial world of its own, and that's so true, because this is what we all are calling microbial ecology. And my question would be, so does the product developer really understand this if they change the chemistry, that they change sometimes the whole world of microbiology?
You're addressing a very good point. If they have a microbiologist in their team, hopefully yes, but often it's not the case. Then you have nice marketing colleagues and let's say process or beverage technologists. They certainly have a basic knowledge about microbiology, but if you go really into new product development, I always would recommend to consult a microbiologist. Yeah.
Okay, thanks.
And that's what often the startups, unfortunately. Every year we are in touch with 4 or 5 startups and only one survives. And often the microbiology is the reason. Then they have bacillus in very sensitive products or any spore forming stuff.
Thank you. Christine, you raised your hand as well.
Yes, thank you. Very fascinating world of microbiology that many of us don't really know about, I guess. I have a question. Normally you say that you find what you look for. And I mean, in many cases, you will not go through a program to look for new things. So probably you know what are the most ubiquitous contaminants, or what do you have to look for at a specific place? Or would you recommend that to get a whole picture or complete picture, that you really should do a lot of more general screening for contaminants?
So if you have a, let's say, quite robust product like beer or like a soft drink, then you use your standard media, you know, that are appropriate for this certain window. In this case, it's very seldom that you have new microbes popping up. But from a hygiene point of view, it's often very good to use then a broad, let's say, approach with different culture media, as we did, for example, in this propagation process. We just took, let's say, 6 other media, and then you had like, I don't know, 10 new species. And it's only a culture media approach. As Dr. Snaidr said, with culture media, it's always only a certain window. But for beer, it's like lactic acid bacteria and some fermentative yeast. For soft drinks, it's often also yeast, lactic acid bacteria, and in low carbonated, like acetic bacteria, then you have your certain windows, yeah, for sure. But as microbiologists, it's really always then very fascinating if you have a new term like this Lactobacillus cerevisiae I showed, and then you can describe it and check which spoilage potential it has. And I have to mention, I'm very happy that Saccharomyces cerevisiae was microbe of the year, I think 2 years ago in your society. So thank you for that, because this is my hobby, Saccharomyces yeast.
Thank you. Well perceived, yeah. Any other question from the audience, either directly or in the chat? And we can also open the panel. We are a little bit, let's say, out of time, but nevertheless, give us 2-3 minutes for maybe also kind of panel discussions also directed to Dr. Snaidr. If there is no, Christine?
I have another question, more probably to Dr. Hutzler. I was fascinated with the picture that you showed on the, let's say, yeast cultivation, when you have this large, I've worked in the yeast industry, and I know that some contaminants are always in yeast, but the amount of the number of contaminants is so low that it's always at a very threshold level how to detect this. How did you go to really find extremely low concentration of contaminants? What is the challenge in that?
So we used then media with either actidione cycloheximide,
Okay.
or a certain copper sulfate
So you
concentration.
differentiate between.
So they suppress then the Saccharomyces yeast, and if you choose the right concentration, then some wild yeast still grow, but without such, let's say, hint, it would be difficult. So suppression.
Actually, if I am allowed to have another question that goes to Dr. Snaidr, I liked your pictures also and your indication that microbiomes, complexes, bacterial societies could also be analyzed very nicely with your technology. What kind of microbiomes have you been looking at, or your partners in companies or academia? Can you give examples on that?
Yes, so at the moment we have looked at different microbiomes, mostly of course in the colon, and in all parts of the colon, but also we have analyzed microbiome in the mouth. We have analyzed microbiome on the skin also. We have some techniques developed to take the samples, and from away from a human, we have also analyzed some microbiomes in animals like mouse or rats, also dogs also too. So it's a fascinating method to analyze the microbiome, and the microbiome itself is really great fascinating.
Thank you.
Okay.
Maybe also one final question from my side to Dr. Snaidr. You were advocating to, let's say, use other methods than cultivation. That was a little bit what I learned. And for somebody who is in the probiotics business, for animal, for human, and you have to give a plate spore count or a CAVU value, what is the next best method of choice beyond the cultivation, if it's about the quantification, not what is contaminating, what is the quantitative, is it flow cytometry, what is your method of choice, what would be your recommendation to do the quantitative step?
Yes, so my recommendation would be, so we are doing a lot of stuff for the probiotic business. So what we recommend there is flow cytometry. On the one side, flow cytometry with a dead, live, or almost or a dead live damage differentiation, but also what we're doing is this fish or a flow fit or fish flow cytometry, where we combine the flow cytometry for the probiotic industry with specific fish probes, and then do the quantification.
Okay.
Stefan, there's one last question maybe in the chat. Can I cite that? Klaus-Peter Keuer would, it's on my side. Klaus-Peter Keuer would like to know, can you, and that goes probably to Dr. Hutzler, can you comment on the time requirements for impurity identification in industry? Because you would like it to have it next day. Does that work?
No.
Clear answer here.
So that's often the challenge we have. So with the direct methods, even if you use centrifugation or anything, and you apply, let's say, real-time PCR, then you have a detection limit, let's say 50 or 100 microbes per ml, for example. And then you have the lag phase, and in conditions like beer or environments that are quite harmful still for the microbe, they have a very long lag phase. So we have made observations that for some beer spoilage bacteria, we need 10 days until we can start the growing process, and that they can be over 50 to 100 cells. And that's the point. So that's when you say, okay, 3 days cultivation, and then you do a downstream method, and then you get a positive results. In my point of view, that's still not feasible. If you find a method where we can really concentrate and check every microbe. And that's maybe the big challenge for Dr. Snaidr, then I would say, yes, you can do a direct real-time product release. And maybe you will find it, hopefully. I cross the fingers.
Thank you very much. Yeah, maybe Christine, is there something in the chat which I can't see still?
No,
or okay,
no, that's fine.
thank you. Thank